Could we create a virus from scratch? It's a question that sounds like it belongs in the plot of a sci-fi thriller. But this is not fiction. This is the edge of reality, the place where biology, chemistry, and information science intersect. The very idea of building a virus, molecule by molecule, gene by gene, unlocks doors to both the potential of healing and the risk of harm. And yet, it all begins in a lab. A quiet room filled with beakers, sequencers, and the humming rhythm of data analysis machines. In such a place, something unseen to the naked eye can be assembled with staggering precision. Synthetic biology is the field where this happens. Unlike traditional virology, which studies viruses that already exist, synthetic biology asks a more radical question. What if we design life from the ground up? And yes, that includes viruses. Not necessarily to cause disease, but to study, to understand, and perhaps even to cure. Let me take you on a journey into how this is possible. First, understand that viruses are not truly living organisms. They are genetic codes wrapped in protein, requiring a host to replicate. This simplicity makes them ideal candidates for synthetic design. All a scientist needs is a blueprint of a viral genome, and the tools to print and assemble it. Back in 2002, a team of researchers made history by synthesizing the poliovirus from scratch. They ordered DNA fragments from a company, stitched them together using enzymes, and eventually produced a working, infectious virus. The scientific world was stunned. It was the first time that an organism had been constructed entirely from synthetic parts. Since then, the technology has evolved rapidly. We now have access to gene printers, CRISPR editing, and DNA synthesis techniques that make viral construction faster, cheaper, and more accurate than ever. But what is the purpose of such creation? One reason is vaccine development. By building a virus synthetically, scientists can create weakened or altered forms that teach our immune systems how to respond, without causing illness. Synthetic viruses have been used to accelerate the production of mRNA vaccines, such as those used against COVID-19. Others are engineered as delivery vehicles, transporting genetic material into cells in gene therapy. Still, every scientific advance comes with ethical shadows. Could a synthetic virus be used as a bioweapon? Could rogue actors order viral DNA online and create something harmful? These concerns are not hypothetical. In fact, the ability to synthesize pathogens has led to a new branch of biosecurity policy. Governments now regulate which sequences can be ordered, and companies screen requests for dangerous genes. It is a delicate balance between enabling research and preventing misuse. But this balance also reveals something deeper about humanity, our desire to control nature, to understand it so thoroughly that we can replicate it. The creation of a synthetic virus is a scientific mirror. It reflects our brilliance and our hubris, our creativity and our caution. And it is not just about creating viruses. It is about designing tools. Synthetic viruses can help us explore cancer treatments, correct genetic disorders, or even build new kinds of vaccines that adjust to evolving diseases. Imagine a future where your immune system is constantly updated like an app, trained to fight off pathogens it has never seen before, all thanks to a custom-designed viral carrier. But here's the twist. Creating a virus from scratch is not just about science. It's about story. Every nucleotide in that virus tells a tale of innovation, risk, and foresight. It's about the researchers who work late nights assembling code from chemicals, the ethicists debating what boundaries should exist, and the governments deciding what rules to enforce. Could we create a virus from scratch? We already have. But should we? That's the more difficult question. As we enter an era of programmable biology, the boundaries between natural and artificial life blur. We are becoming authors of microscopic narratives that can change the world. The deeper you go into synthetic biology, the more you realize it's not just a field of science, it's a philosophy. What is life? What should we do with the power to rewrite it? And can we truly foresee the consequences of our own creation? One thing is certain. The tools now exist. The possibility is real. What remains is the wisdom to use them with care. The next great pandemic could be stopped by a synthetic virus designed to train our immune systems before danger strikes. Or it could begin with an unregulated lab somewhere in the world. The outcome depends on vigilance, ethics, and global cooperation. So, the next time you hear the phrase, "created in a lab," remember it's not always a conspiracy theory. Sometimes, it's a glimpse into the future of medicine, evolution, and how we relate to life itself. And that story is still being written, one gene at a time.